So thank you, Juanma, and please.
Thank you very much to you and all of you. I try to see the presentation. Do you see it?
Yes, perfectly fine.
Okay. As Marta said, I'm going to speak about the different methods to obtain the small vertebrate remains in quaternary sites. We are going to see the presentation in different parts. What are the small mammals, which are the main groups and the species, how we recorded them, and how they arrived to archaeopaleontological sites, and why we studied them. First, what are small mammals? Okay, small mammals are normally, not in a scientific terminology, are the mammals smaller than a rabbit. For example, here you can see different little mammals, but for example, dogs or cats or this little monkey are not small mammals. However, for example, here the beavers or capybara are rodents and are considered the small mammals. Also, they are big. They are big like a dog or something like this. Okay. Here, only a slide with the phylogeny of the Eutherian mammals. We are going to speak about rodents that are here in the phylogeny. After insectivores or order Eulipotifla that comprise basically shrews, hedgehogs, and moles. And after Chiroptera that are bats. And as you see here, the fish that which are separated by the Eulipotifla is the order, the ancient order, after bats, and then rodents. And also here a little bit the morphology of teeth of this group of mammals in general. Rodents comprise the 42% of the mammal species in the world. Also, bats are very, very abundant. They have these small mammals high reproduction rates, as evolving group, a small size, short migrations, specific climatic and environmental requirements, and a wide range geographical distribution. Is that why they are important to biochronological reconstructions in archaeopaleontological sites and are optimal for paleoecological proxies in local and regional scale? We will see in advance. Okay, here only a picture with the screening. You can see in fast how to recover the sediments in an archaeological site. This is a video of Atapuerca. Which are the main groups and species? A little bit. Here, in general, these mammals, there's class Mammalia, we separate 3 orders, Eulipotifla, with the 3 great families and subfamilies, where you can find Erinaceus here, or the small shrews or moles. Also bats, that you have 2 big families. There are a lot of more, but there are basically the remains that you can find in an archaeological site, the Rhinolophidae and Vespertilidae. After rodents, the most abundant in this kind of data are the mice, voles, and hamsters. Also the dormouses. This is the definition of insectivore, the Eulipotifla. It's a small mammal with a small size, with acute molars and tooth and voids and feed insects, and where includes shrews, moles, and hedgehogs. This is the definition of the Dictionary of Zoology. The insectivores evolved quickly 102 million years ago, and the order Oligotyphla appears little just time before the extinction of the dinosaurs, the Catelinid, in 65 million years ago. And the main families of subfamilies that are extant today appears during the Eocene period. Okay. Okay. This is only pictures to you have to see here the teeth or lower molars of this kind of insectivores. Also the upper molars, the morphology, mandibles, for example, here on a cranium, another mandible, and canine mandible. And if you see here this picture that is a cranium of a mole, that you can see here the morphology of the cranium, also the teeth. Are characteristic of this kind of morphology in W, as it's called the ambodont. Also here, you can see the mandible, in this case of an Erinaceus, with all the incisors, incisives, canine, premolar, and molars here. And also other important things like in moles, the arm is important to recognize the species because they are very adapted to excavate. And we have here the humerus, ulna, radius in green, the metacarps, and the phalanges that are diagnostic to recognize almost the genus and maybe sometimes the species. Okay, the other groups are chiropters, that are these mammals that are flying, at the real flying with wings, with a long membrane. We have here the pictures of the phalanges. This is the phalanges of the chiropters that make the wings. Okay, and also the skeleton. You can see here the different parts. It's always the definition of the Dictionary of Zoology. The bats, the really bats, or the bats that we have known nowadays, appears in the Eocene around 52.5 million years ago, and are probably evolved from a fossil arborean small mammal. Here, a little bit the phylogeny of the ancient groups of bats. Also, here the morphology is similar to the insectivores, but have a little bit differences in the morphology in teeth, in cusps, and also in enamel and something. This is a lower molar in occlusal view, and lower molar in buccal view, and an upper molar in occlusal view. Here a mandible and a cranium. You can see here the canine and the mandible of a rhinolophus, with the characteristics of the mandibles with a prolonged canines, an acutate also cusp of molars, more or less. And here only the mandible of an myotis. This is the molars, premolars, the canine, and incisives. It depends on the species. They have more or less premolars. Okay. Also, in bats, it's very important to recognize to a genus or a species level the distal epiphysis of the humerus. For example, this is the different views of an humerus of the genus Myotis. Here, the differences between the humerus of Rhinolophus species and other species that we can find in archaeological sites like Miniopterus or Barbastella or some Nyctalus. Do you see here the differences are clear? After recognize the species, the genus or species level, it depends on the material that you find. Finally, this is the definition of dictionary of zoology of the rodents. See, this is and you can see all the skeleton of a rodent and the different species that include in this order. Here there is a beaver eating a tree here. Okay. These rodents, the oldest fossil we found in Africa in 52 million years ago, more or less, in the late Pliocene, sorry. And here, more or less, the simplified phylogeny of these species. Rodents are characteristic. This is a cranium. This is the ventral view or the occlusal view with teeth and lateral views. The main characteristic that they have not canines. They are incisors. They are here in a hole that it's called diastema. And the smallest species, there are only molars, not have premolars. Here, the biggest species maybe have 1 P3 or P4, but the smallest species have only molars. You can see here a cranium of a rodent with the incisive here. And here you can see more or less 3 molars. And this hole between the incisives and the molars that is called diastema. It's the same thing for mandibles. They have the incisives, the diastema, and after a third molar. 1, 2, 3. Okay, this is inside the order Rodentia or the order of rodents. This is the most abundant species, more or less, or groups that we can find in archaeological sites. For example, bowls, their teeth, the occlusal view or bowl's teeth, and a picture of the current species. Also, mice that you can find this morphology of teeth is different. These are upper molars and lower molars, and also dormouses that are here. Lower dentition. For example, these are exception, but dormouses are little rodents or small rodents that have also a before lower before. But it's not general. For example, here the mice or mouse have only this is the first molar and second molar, but they don't have a before. Okay. Slide. Hello.
Okay.
Okay. Yeah. How we recovered them? The basic analytical techniques to obtain these small pieces of bones and teeth in archaeological sites. The first one is the samples collection. The sediments are picked from the excavation and pointed in as samples. After there was the water sieving or water screening with different type of measures. After this sediment that contains also also the bones and teeth are picked up to obtain these small vertebrate or small mammal remains, and after they exist the identification with microscopes and other techniques, and the quantification with different kind of methods of number of identified specimens or number of remains, minimum number of individuals. Also here, the different pictures from Atapuerca Norte, with the different methods and water skinning methods with different kind of meshes with different millimeters. Here the drones, and here the pictures with this methodology. Sorting and preparation for the analysis. We have the material. After also with this material, when we pick up and we separate the identified material, but because it depends on groups, there are groups that on small mammals that you can identify only teeth. There are other groups when you can identify other kind of bones. After, for example, there are other kind of small vertebrate like amphibians or reptiles that you can identify some other bones, not only teeth, for example, or birds. And we have normally a reference collection with career material to compare it. You can find here, for example, even as a red-toothed shrew, and this is here an a bat cranium or a mandible of the reference collection, and this is the identification with the microscope that you can take measurements, or more advanced pictures and techniques for after analysis are the ESM, the electronical environmental microscope. Another kind of thing is how they arrive to these archaeological and paleontological sites. Normally, there are different kind of predators that accumulate these small mammals in a site. Are the owls or nocturnal birds of prey, diurnal raptors or diurnal birds of prey, small carnivores, and sometimes other agents like humans, for example. This is a drawing on how they arrive to the sediments in a cave. And this is important to know the information of the environment and a good taxonomic study. This is a list of more or less of nocturnal birds of prey that are common accumulators of small mammals in caves and that we can find in archaeological or paleontological sites. And this is the method of the accumulation of these kind of predators. They bring the prey entire, and after make the regurgitation of the parts that they are not digestive for them, like bones and hair or other parts that are not meat for them. And you can find in these things that are called pellets the different kind of bones of, for example, small mammals, and this is clean. The different mandibles here, a cranium, also here a femur, the different parts of small mammals that you can recover in these pellets. This is the parts of the formation of a pellet. Here in this video, you can see how these nocturnal birds of prey make the regurgitation of this pellet is something like this. Okay. And in taking into account the predator, there are different categories. So, for example, I'm going to speak of mainly in digestion, that are different types of digestion and category of predator in relation of the bones digested that you can find. The digestion is normally studied in incisors, molars, and femurs. You separate absent, light, moderate, heavy, and extreme. After, there are other kind of separations that we can make us identify the predator as the skeletal representation that we found, or the type of breakage of in molars or in other bones. As example, we can find here the different types of digestion, from moderate to extreme. This is mandibles and teeth of a red tooth root here, where is marked the moderate digestion here. Heavy digestion, you can see here, that are more intensive, and the extended digestion in the same species, that is completely different. In fact, the red pigmentation in teeth is not shown in extreme digestion. After here, light, moderate, heavy to extreme, also for the arbiculine teeth, the bowls teeth, you can see here, also perfectly the triangles of teeth in light, less better in moderate, and in extreme and heavy and extreme, the teeth are completely broken. Oh, pass it away. And also the differences between the light, moderate, heavy, and extreme digestion in incisors, that is very studied in the small mammals taxonomy. Okay. In other cases, there are other things that affect the bones, like water abrasion here, or roots, or oxide of manganese oxide, that can affect the composition of bones. Okay, why we study these small mammals in this case, or small vertebrates? Different approaches, like biochronology, paleoecology, and paleoenvironment, or biological evolution history, or evolutionary models. I'm going to speak here only about biochronology and paleoecology and paleoenvironment, that are the three main research lines that you can find in archaeological or paleontological sites. Also, biological evolutionary story, and this kind of evolutionary models, now it's in the top for the ancient DNA studies in rodents, in both that are last year's published. But I focus in biochronology and paleoecology. Okay. To understand the biochronology, it's important in small mammals, mainly in rodents, because he have high reproductive taxes and fast evolutionary rates. It's an example, for example, of the Coca-Cola, no? with all these years that change quickly. Also, this kind of rodents in the Quaternary evolve quickly. The Biochronology, as definition, is a part of paleontology that study when paleobiological entities lived, and in temporal order and the dating of past biotic events. This, for summarize, is the one you can more or less make a chronological range of a site when you have determined small mammals or rodents association in this site in relation to other sequences. Okay. This is the period of the evolution. I make this chronostratigraphic chart because, if you can see, Quaternary is a small period, and this kind of small mammals evolve really very quickly in a little short period of time. Okay, the most used or useful rodents in this case for these biochronological studies are bicoids, are voles, that are a subfamily of rodents that includes voles, water voles, lemmings, and muskrats. This is a common vole, the water vole here, or a Norway lemming, and this muskrat as the species that compose this subfamily of rodents. This origin is in the around 5.5 million years ago in North America, and in Poland we can find this kind of molars that are not really arbicolids. This is an arbicolid. This kind of voles evolved from hamsters, from a form of Cricetus. These tubercles of Cricetus are elongate and conform these different types of triangles. And we can find here the first species that is recognized as arbicolid, as an arbicolid, Phomimomes insuliferus, that provides from Poland. To make more understandable these things, I make some examples. For example, for the biochronology, I'm going to speak this example that we published in 2015 about the chronological context of Pirro North with the sarbicoid species like Alphaiomis or Pirro North. Marta knows better than me and other Italian guys. It's a fossil deposit located in near town of Abrichena. This is this location. This is a quarry. Here is Pirro 13, where they found lithic tools. There's the picture of the quarry. Here a general picture, and here the people water skinning the sediments in this little swamp, or I don't know how it's said, because it's not a lake, it's only a stream stagnant water. Okay, and because
Let us call it lake, please.
It's a lake.
For us, yes.
Okay, it's a little lake. Okay, in Pirro we identify this small mammal association, and it's important the identification of this species. That this is 20 rounds that we've done for this paper. Are all the same species with the different morphology, the morphological variation of the same species. This species is Alphaiomis. We compare this Alphaiomis from Pirro North with other Alphaiomis from Italy and other ancient species from other sites in Europe, also with the ancient Alphaiomis from the lower layers of Cima Elefante and Grandolina. And we shown that are separated from these ones. Here is Pirro, for example, the Alphaiomis labocati from Elefante, and other sites are here, appears here, or here the labocati. And we conclude more or less for like this. You can see why is useful for biochronology, more or less, that this Pirro 13 has a chronology between 1.3 and 1.6 million years that is older than other layers from the lower layers of Atapuerca, and also from lower layers of Barranco León in North. But you can see here. Another point of view for paleoecology and paleoenvironment, It's only a little scheme of a colleague that make about different kind of rodents that lived or need. They are very specific documents, some species like hamsters or microtus or other type of voles or also dormouses that needs an environment and climatic requirements very specific to live. And is one of the things because they are very important to reconstruct the environment or the climate because depends on the assemblage that you find. You can find one kind of habitat or other kind of habitat or one type of climate or other type. Okay. Here only for make an example, this is the current landscape in Sierra Atapuerca 1 million years ago when there are one antecessor. And for example, this is a reconstruction of the landscape from Sierra Atapuerca that is very different, very different from nowadays. Also, other side is in Asturias, in northern Iberia. This is the current landscape in Asturias and the reconstruction for where Neanderthals living there in 40 kilo years ago. Okay, this is only an example summarized of the climate of the Quaternary fluctuations from 2.0 to 0 more or less. In blue, the glacial periods, in yellow, interglacial periods, with the different marine isotopic stages. MIS is marine isotopic stages. The pair numbers are called periods normally, and the odd numbers are warm periods. In general, because after inside the warm periods or cold periods, they are cold or warm fluctuations inside. This is only a general. Okay. Different methods what we use normally to reconstruct the environment and climate with these small mammals or small vertebrates. For example, habitat weighting method that consists in the repartition of the different small mammal species is an example from another publication where you can find here insectivores, bats, and rodents are there in different kind of method to simplify after the interpretation, open dry, open humid, woodland, rocky, or water. Another method that we use normally to make this kind of interpretation is the bioclimatic model. That is the more or less the same idea of the habitat weighting, but the repartition in habitats is not here, is in types of climate. So it's another example with water species divided in different type of climates, or like subtropical, typical temperate, arid temperate, cold temperate, or polar. After with linear regressions, we can calculate mean annual temperatures, mean temperatures of warmest months, of coldest months, and mean annual precipitations. Another method that we use to reconstruct the climate is the mutual eco-geographic range that consists in see where coincide with or a small mammal or small vertebrate association in different points, and after overlap this intersection with maps of current temperatures of precipitations to make the calculations of temperatures or precipitations. Here is an example of two species. This is the Iberian vole with the common vole. This is the distribution, current distribution in the Iberian Peninsula. This is the intersection. The intersection is where coincide these two species. And here the overlap with a climatic map. I think that is from mean annual temperatures in 30 years. And this is another example with the reconstruction of a site of Atapuerca. Is the Portland site, the Pleistocene site, with the different, the red line is are the current values from mean annual temperature. This is warmest month, coldest month, and because it's in Spanish, I'm sorry, and mean annual precipitation, and the different values that we obtained with the small mammal assemblages. For example, we can see here that the values are lower from current ones. I don't remember exactly, but I think that, for example, it's coincide more or less with the Henry 7.3, that is a cold peak in the marine isotopic stage 3. And as an example of these reconstructions, I put this example of this publication that we made about the Fumane cave, this cave that is in Verona in Italy in 2015 also, that has a good succession of marine isotopic stage 3. Grotta Fumani is located at 350 meters in western part of Monte Lessini. We can hear a picture of the map of Italy. Here the picture of the site. Here is the stratigraphy, and I marked here the levels that we study, where are Neanderthals and atomic modern humans occupation, and also is where the small vertebrates are recovered. Okay, these are the different radiocarbon dates from D1 to A5 plus A6, and the calibration with the GSP 18 oxygen. And more or less it is considered from 50 kilo years to 35 kilo years. The fossil assemblage that we found in Fumane cave. I'm sorry, I have a cat. Calla. Sorry. She's angry. I think, no, I don't know. The fossil assemblage include 8,657 identified specimens. We have here insectivore from Einaceus to Neomis, after bats, and after rodents. That corresponds more or less to 4,340 individuals and 29 taxa. There is a lot of material. The most interesting species in all these sequence are, for example, the northern bone, Chienomis nivalis. This species here is a tooth. Cricetus is a hamster, this teeth here. Dinaromis bogdanovi, that is a next species, a Balkan bowl. This is the Balkan bowl. And for example, Microturus economus is the northern bowl. That is this tooth here. And these are the drones of different kind of species of bats and insectivores that we identified. Finally, the most important thing, and this is the reconstruction of the landscape with the habitat weighting method, open, with land, rocky water, the calculations of different climatic parameters, temperatures, and the diversity. We can see the different changes. But finally, in general, the most important thing is that more or less both in the layers where Neanderthals live, in the Mousterian layers and Aurignacian layers, are well adapted to the environment because there are cold moments here and warm here, or two warm here and one cold here also. And to finalize a little bit, I'm going to speak a little bit about new techniques and improvement that are these last years are applied to the small mammals studies. For example, this is from the the PhD thesis of Mónica Fernández Garcia. This was my PhD student that used the incisive of rodents to make oxygen isotopic analysis to calculate the ratio of the oxygen 18-16 in rodent teeth and compare with other methods. For example, this is the comparison with mutual ecogeographic range and bioclimatic model with different sites. This is a site in northeastern Iberian Peninsula. I'm sorry, one moment, please. Sí, la puerta. Sorry. This is the reconstruction of the Arbreda cave and Teixoneras together, two different sites, and also this is the reconstruction with the oxygen isotope from rodents. And this is another site. As you see, in general, there are the tendency as more or less the same, but after the points of temperatures and precipitations are different, but because it's normally in relation of the methods that you use for that. But more or less are the same data that you can find with other classical methods as mutual ecogeographic range or bioclimatic model. In this case, it's only for the climatic reconstruction, the comparison of the climatic reconstruction with oxygen isotopes and other kind of methods. Another improvement in this kind of method is the application of GIS to mutual ecogeographic range method. In this case, is the PhD from Manazawaga in 2020 apply GIS to make the mutual ecogeographic range and calculate the temperatures and precipitations of the site of El Sal that is located in Valencia, and is a MIS3 site in this case. And we have here also the habitat weighting and comparison of the different kind of temperatures and precipitations. Also, another kind of method that are applying now for the study of small animals, in this case for the very identification of some species, for example, morphometric morphology. These are the PhD thesis from another of my students, Ivan Rey Rodriguez, in 2021. This is an example of the manuscript that he published about one species of this kind of bowls that is found in Iran, and the comparison of the different species and the morphometry that helps to separate some of these species one from another. And finally, another kind of words that we can find now is DNA. This is a study that we made in collaboration with Poland people back at 2020. This is only the separation of two bowl species, the common bowl and the field bowl. This kind of studies is useful to know the origin of the different kind of species. For example, this is the repartition more or less of the different lineages of the species currently in Europe. You can find, for example, here that the origin of the western north species, for example, here in green is here, and is this part of North Europe, or the origin of this expansion of these Microtus currently species the Iberian Peninsula, for example, or the origin of the yellow points that Central Europe are really Central Europe. Okay. And I think that that's all. I don't know what time is it, but thank you for your attention. And if you have any